After watching Professor Dave and glycolysis and all the actual steps that occur, now you can see why I let him explain it, not me. There's a lot of chemical things going on there. But again, this is biology. So the big thing I want you to do is realize that there's a lot of steps that take place, but we're not going to go through every one of these because this isn't an organic or a microbiology class. This is regular biology. Okay, so after it goes through glycolysis, then it goes into the acetyl-CoA, or another name for it is the Krebs cycle. And again, you can see there's a lot of stuff going on. All of these steps are helped by enzymes. We've got NADH is another chemical compound that where you get bonds being transferred. Here's an ADP turning to an ATP. So a few happen here. But again, what we're really doing here is we're taking that glucose and we're really breaking it down so it's ready for that last cycle. Okay, so the citric acid cycle actually has 8 steps, and each one of them has a specific enzyme. And again, we talked about enzymes. They all are very, very, very specific. This enzyme is, okay? So the acetyl-CoA joins the cycle. It combines with what they call oxaloacetyl. So it's acetate with an oxygen, and it forms citrate. And now that's the first step. The next 7 steps is to really take that citrate and break it down. Okay, so it's a cycle. It takes 8 steps, and basically what it really is doing is really breaking down. We took glucose and we turned it into pyruvate, and then we added some CoA to it. We added an enzyme to it, and then we throw it in the Krebs cycle. When we're all done, that glucose is broken down. Okay, the NADH and the FADH, those are part of the cycle. They help with the electron. Just remember that, they're formed there. I'm not going to get big time into it because again, it's a lot of chemistry and we don't really need to get into that much. But here, I don't know if you can see the word electron here, but electron transport chain, that's what we used to call this last cycle. Now they've changed the name, but they call it oxidative phosphorylation. And again, we're adding those phosphate groups, so maybe that's where it came from. Electron transport was way easier to remember. But this is the last step. Okay, so now we're really ready to put that. We've got a couple ATPs, one here and one here formed. But this is where they really get formed. This is really where we get our energy. Okay, so during oxidative phosphorylation, there's a chemosymosis. Okay, what happens here is we're going to transfer electrons and we're going to change ADP into ATP. That's where we're going to get that high energy bond, and we're going to make these ATPs. And the energy is extracted from the food, the glucose that you got when you ate. Okay, there's 2 electron carriers, and they donate electrons to this chain, and that's where you get the power to change ADP into ATP. Okay, that's what happens in the oxidative phosphorylation or the electron transport. This is where we really get the ATPs formed. Now, if you don't have oxygen, this step isn't going to happen. Okay, so if you go through anaerobic, you only get those couple ATP formed from like the glycolysis and from the Krebs cycle, just those 2. When you get to this one, you get a bunch more. Okay, so the pathway to the electron transport is in the inner membrane. The mitochondria has a double membrane. The outside's smooth, the inside's all jagged. Okay, and those little folds are called cristae, and this is where it happens. Okay, most of the chain's components are proteins, and they have multi-protein complexes. So you've got a lot of stuff going on in those cristae, but really what they are is carriers. They're going to reduce and oxidize, they're going to accept and donate protons, and eventually you're going to add those phosphate groups onto that ADP. You get ATP, that's where we get our energy. Okay, the electrons, again, drop in free energy, and that's what happens. And they finally pass to the oxygen, and they form H2O, and then that's where you get your energy coming from. Okay, so the electron transport system is the one, excuse me, where most of the ATP is formed. And basically, you're adding that high energy bond onto ADP, and that's where we get our ATP from. Okay, so here's the whole process. Glycolysis is taking glucose and breaking it down. Okay, and then it goes into this where it becomes what they call pyruvate. It's starting to break it down, but it's not quite ready to be broken down all the way. There's a little step in here that they didn't say, where that's the acetyl-CoA, where we add a coenzyme. Then it goes through the citric acid cycle or the Krebs cycle, and that really breaks it down. That totally breaks it down into, and then we can throw it in here, the oxidative phosphorylation, like I said, electron transport, and that's where we get most of our ATP. So we get 1 ATP here, 1 ATP, and I think we get 30 from this one. Okay, so during cellular respiration, most of the energy flows in this sequence. It goes from glucose, and then NADH, then electron transport chain, and then proton, motive, force, in other words, adding that ATP. Okay, that's the kind of the chain that happens. Okay, about 34% of the energy in glucose molecule is transferred to ATP. Okay, so we get, it's not, you know, super efficient. So that means we get to eat more. Yay, right? Okay, so it transfers 34% of its energy into making that ATP during cellular respiration. And again, we get about 32. So 1 from glycolysis, 1 from the Krebs cycle, and 30 from the ETS, from the oxidative phosphorylation. I'm probably going to call it electron transport, you guys, because that's how I learned it. But again, there's reasons why we don't know exactly. They say 32, but it might be more, it might be less. Some of the reasons is, you know, that's a pretty small place for everything's to happen. So it's an estimation. Okay, we talked about that's all with air. Okay, cellular respiration, that's occurring, that's aerobic. Okay, but remember we said it can happen without oxygen. There's fermentation and there's anaerobic respiration. Okay, they produce some ATP without oxygen, not a lot though. Okay, most of it requires oxygen. Okay, you're going to get a couple ATPs without oxygen, but without oxygen, you don't get that electron transport, that big one at the end, and that's where you get most of your ATP. So the electron transport chain won't happen. You won't do the last step. You'll just do the first ones. And so you're only going to get 2 ATP. Okay, so glycolysis coupled with anaerobic respiration or fermentation, it does produce ATP, but it only produces 2 molecules of it. Okay. All right, so it uses a different system. It uses a final electron acceptor other than oxygen. It might use sulfate. It might use another one it uses is something called lactic acid. So there's something different going on besides accepting oxygen. And because it uses that, it produces a different form. Okay, so when we're talking about fermentation, instead of the electron transport chain to generate it, it uses a different thing, a substrate level. It's going to occur in a different area, and it's going to, you know, we talked about it being oxidative phosphorylation. It's going to be something different here. It's not going to use the oxygen. Okay, so types of fermentation. There's 2 common types of fermentation. There's alcohol fermentation and there's lactic acid. And any of you guys that haven't worked out for a while and then work out pretty hard know about lactic acid. Okay, if there's not enough oxygen in your muscles and stuff, you can't burn, you know, glucose the way you should. So it produces lactic acid, and that's the stuff that makes you really sore. Okay. In alcohol fermentation, we use this a lot because we like our, you know, our beer and our ethanol. Ethanol is all liquor. Okay. What happens here, the pyruvate, which we converted into, you know, put it in the Krebs cycle and the ATS, that doesn't happen. Okay, it releases carbon dioxide, and then the second step, instead of producing a bunch of ATP, produces ethanol. Okay, now this doesn't happen much in our cells. It happens in yeast cells and bacteria cells and stuff like that. And we use that process. If anybody of you have ever made wine or beer or whatever, we use that process. We also use it like yeast produce carbon dioxide when you're baking. You put yeast in there and you want your bread to be light and airy. Well, that's what the yeast does. It releases CO2, which is captured into the matrix of the flour, and that causes it to rise. Okay, so alcohol fermentation by yeast is used in brewing. It's used in winemaking. It's used in baking. There's all kinds of stuff that you use alcohol fermentation for. Okay, so, and again, it's mostly that, you know, making stuff here. You use different kinds of yeast for different things. There's baker's yeast and there's brewer's yeast, and they each have a different job to do, but they release, we use yeast for this part, but if you keep going, then you'll end up making ethanol, which is liquor. Okay. In lactic acid fermentation, again, the pyruvate is reduced. Instead of forming your ATP, you form lactate. That's your end product. So you don't get the release of CO2, but you get this lactic acid. And that's, like I said, what occurs in your muscles. It makes you super sore, especially, you know, it takes a day to make it. And then the next day it's like you can barely get out of bed and everything hurts to move. That's because you didn't have enough O2 there and you produce lactic acid instead of ATP. Okay. There is some lactic acid. Yogurt uses it. You know, they always say about yeast cultures and stuff, or yogurt cultures. When you look at the ingredients, these guys use lactic acid to make cheese, yogurt. So we use lactic acid in other things too. Mostly fungus and bacteria will do lactic acid fermentation. We can do lactic acid fermentation if you want to work really hard after you haven't worked out much. You can make your own lactic acid. Not a fun thing. Okay. Human muscle cells use it to generate ATP when O2 is scarce, but again, you got to get rid of all that lactic acid before the soreness goes away. Okay. The funny thing about it is you work out, you get super sore, so you got lactic acid in your muscles, and the only way to get rid of it is to keep working out. Yeah. So once you work out and you've punished yourself to the point where you're sore, you got to keep working out to get rid of the lactic acid. Oops, sorry. Cellular respiration, again, produces 32 ATP per glucose molecule. So every glucose molecule produces 32. That's when it goes through the last step, the ETS. Fermentation produces only 2. So if you don't have oxygen, you only end up with 2. And again, that's from the first 2, the glycolysis and the Krebs. It doesn't go through the last cycle, and that's why we don't get a lot of ATP. Okay. There are different kind of critters that actually carry out fermentation, carry out anaerobic. And if you put oxygen there, they will not survive. Okay. They're obligate anaerobes. Okay. These are going to be bacteria and certain kinds of fungus and things like that, but it's mostly bacteria. If you put oxygen there, they'll die. So sometimes that's what we use. We expose things to oxygen to kill them. If it's a bacteria that you don't want around and it's an anaerobic, just give it oxygen. Okay. Yeast and many bacteria, they're called facilitative anaerobes, meaning they can survive either way. They can go through fermentation and be fine. You can add oxygen to them and they'll be fine as well. So you've got different things. You've got the ones that have to be anaerobe, obligate, and then you have facilitative. Either way, we can do it with or without. Okay. In a facilitative, the pyruvic, that thing we break glucose down into, the pyruvate, it'll go either way. Once it hits that, it can either go into, it has 2 pathways it can go. Okay. So they have options there, oxygen or no oxygen, whereas these guys don't. These guys only have the one pathway because they don't have oxygen. Okay. And again, you can see it here, the glucose, and we have the pyruvate. That's the normal thing. We break glucose down. It can either go into the mitochondria, go through the whole thing, acetyl-CoA, citric acid, the Krebs cycle, whatever, and then here. Or they can just go right down into producing ethanol and lactate. So they've got options, okay? If there's oxygen present, they go here. If there isn't, they go here. Okay? So why is glycolysis so important? Well, the ancient prokaryotes, and we had prokaryotes before we had eukaryotes, they used glycolysis before there was oxygen. At one point, our atmosphere didn't have much oxygen in it. Oxygen was actually a, what you call a pollutant. Okay, it was more methane and carbon dioxide and things like that. But once things started going through photosynthesis, they started adding oxygen. So they totally started to change our atmosphere. So the first guys didn't have much oxygen, so they had to use whatever was available. So the early prokaryotes, the early, the simple cells, used only glycolysis. That's all they had. They didn't go through the next steps. They didn't do the Krebs cycle. They didn't do the ETS. They didn't have that option. Okay, so they generated ATP just through glycolysis. So they only got 1 ATP every time they broke down a glucose molecule. Okay, so it's a very ancient process, but it's been around a long time. It's the beginning of the process for cellular respiration, but for some things, it was the beginning and the end. And speaking of end, we're at our 15 minutes, so this is where I'm going to quit for the day.